in the symptom complex. The clinical evolution of disease occurs over years, and so your outcomes kind of have to be very long-term. There are no validated biomarkers, there are no validated outcome measures, and the dosing is even unclear. If you look at that little mouse brain up there, it's pretty easy to get drug into it, and how do we adapt that to the very large human brain? And this is just an example from Niemann-Pick type C, where you can see our severity scale. When it goes up, the patients progress, but these patients progress often over decades. So it makes it very hard to study this in a clinical trial setting. So this is the basic problem. We have historically done clinical trials for a clinical problem that has a plethora of genetics. And now we have single gene problems that we're attacking, and there are multiple clinical problems. And this is a fundamentally different problem from the standard clinical trials for clinical problems that we've done for ages, and is going to require new outcome strategies and trial designs. So what are some of the design issues for each rare disease trial? Should you have an open label or placebo control? How long should the placebo be? Are there enough patients for the stats to even make sense in a placebo-controlled trial? Are the patients losing function, and it would be unethical to put them on placebo for the length of time it would take to see a benefit from the drug? Are there multiple drugs that could be used for the same disease? Are there multiple diseases that could benefit from the same drug? Can we use some kind of novel trial design to approach that? How do we figure out the dosing and translate from preclinical? Is it the same as if there's no disorder? Should we use multiple ascending dose initially? Should it be within person or in cohorts? Safety assessments, oftentimes the participants are impaired and they can't report adverse events. How do we monitor that? The age of the subjects. Often we think younger patients or patients earlier in disease would benefit the most, but then they don't maybe have enough symptoms to track, or there's safety issues with starting with kids. And then the formulation of the drug. How are we delivering it? If we need to put it in the spinal fluid, how are we getting it there? With an LP, in the ventricles, in the cisterna magna, what's the best way to do it? And then the outcome measures, which are often a huge problem. The distance that patients have to travel to get treatment in a rare disease if the study only runs at a few centers. And so your outcome measures, though, are going to depend on whether it's placebo-controlled or not, because things have to be a little more objective with a placebo-controlled trial. So there are a couple of novel designs that have been proposed, like the basket trial, where you can look at multiple rare diseases with a similar pathophysiology and try to treat them with a single agent, and then you can combine data to get better power. The umbrella trial, where you have multiple drugs for a rare disease. Angelman syndrome is a great example of this. 3 ASOs in development, all using basically the same measures. You could combine all 3 trials and just have one placebo group. The problem is that you have to get sponsors to work together then, and that doesn't always happen, or doesn't ever happen. And then the platform trial, where you can have multiple different interventions for the same disease, and then you can have an adaptive design where you throw something out if it's not working. This requires your outcome to be something like a biomarker or a rapid clinical outcome. You can't use things that take years to demonstrate benefit in an adaptive design like that. So outcome measures. What are our big hurdles in outcome measures? Well, we often need to test a broad functional range, and the higher and lower groups fall off and have ceiling or floor effects. There are very few measures that cross the entire range of functioning, and few are validated for special populations like intellectual disability. It's not clear how you get at some of the core defects because there's no measure developed for those. Sometimes the patient can't actually cooperate for the outcome measure to be delivered. And based on the current model, we have to guess one primary outcome in a genetic disease with many problems, and then FDA approval can hinge on this. Composites have not been well accepted at FDA, so we often need to design special measures or special approaches to use measures like disease-specific clinician global impressions. And so this is always a conundrum, is how do we accept something, how do we find something that will be acceptable to FDA, but will also capture the disease? And so this is just another slide about the historical approach, and this I showed at a FDA meeting on epilepsy. and neurodevelopmental disorders. And so when we have a disorder that involves seizures, it's very simple. We can measure seizures, and then, or something like ADHD, or we have one symptom. When we have a genetic disorder that causes cognitive and brain dysfunction, general brain dysfunction, we have all these outcomes. And so this is most of our genetic diseases, where we're going to have all these different problems, and we don't have a way of measuring across all those problems. So once again, we've spent a lot of time, especially in the Fragile X field, we've spent 20 years trying to pound a square peg into a round hole. So what might we consider for multi-domain outcome measures? And I'm just going to kind of go over this in general, because we're going to have more on that later. But we could develop a disease rating scale incorporating the key problems in the disease. We could make a CGI of severity and improvement with anchored domains that are important in the disease, and we could involve families in the development of those domains. We could have a multi-domain responder index where we take different measures and combine response on those different measures that assess different parts of the disease. We could have a composite of known measures that cover key problems in the disease, or we could have a family of outcomes where improvement on any of the outcomes counts as a responder. And these have all been used in different trials that are listed at the bottom, but they, again, are kind of a high bar to get acceptance. And endpoints in genetic conditions with intellectual disability are an even bigger problem. Why? Because the tests were not created or standardized for intellectually disabled populations. Their patients have floored test scores because you're comparing them to normal development in the standard scores. We need to use growth score values or person ability scores, which tell you kind of absolutely what is that person's ability and not what is their ability compared to a normally developing person. We need to use tests out of age range, because if your patient has a cognitive age has a mental age of 3, you can't use a normal IQ test. And, you know, again, the GSVs, the variation in ability level within the same disease may actually prevent you from even using the same measure in all of the patients with the disease. And then the tests may not even be granular enough to detect important relative improvements in the neurodevelopmental disability, even if you use growth score values. And this is an example of that. We actually reviewed like 900 CGI interviews for patients in the 3 Angelman trials, of which we currently have about 80 patients on an ASO for Angelman syndrome. And we asked, when do the Bailey and Vineland, Bailey is a developmental test for kids under age 3.5 that we use in Angelman syndrome to detect improvement in developmental function, and the Vineland is an adaptive behavior scale. And it turns out that only about 23% of what the parents report to us is detected on the Bailey and the Vineland. And these are our primary outcomes for these trials, for phase 3 trials at the FDA. So what's an example of that? Well, if you have an Angelman patient who can walk 10 steps, they get maximum points on the Bailey for that. If that patient will only walk a block and then insists on having their giant stroller that holds a 14-year-old, then that's a bit of a burden on the family. If that patient gets improved stamina and balance, and now can walk indefinitely, and goes on a trip to Africa without the stroller and walks all over Africa, that gets you 0 points on these measures. Patients with Angelman syndrome love water. They will walk into water and drown if they can't hold their breath. If you have a patient with Angelman syndrome in a trial who starts holding their breath and learns to swim across an Olympic-sized pool, which is a significant safety improvement, that gets you 0 points on these measures. So this is just an example of our outcome measure problem in these diseases. The second big problem, as I see it in rare diseases, is the control group in trial design. There may not be enough patients for a statistically meaningful control group. Manifestations in different patients may be very heterogeneous. We can get different groups randomized to the placebo group in a small trial, and then basically we call that randomization roulette, and I took that from the muscular dystrophy field, but you can get a control group that's just different from your placebo group just by chance, because there's a lot of variability. Matched natural history patients may actually be a more accurate control group, because then you're matching people so they are the same. And patient as their own control is another and possibly the best way of knowing whether a patient has changed on a disease, and we need to have statistical models for that. It can take years to see outcome, and we can't put people on placebo for years. And there's often an unmet need with a highly severe disease, and we have a genetic treatment that might work, but delays in designing these kinds of studies can delay access to an effective drug. And so this is the patient has own control. You need some lead-in natural history. So if you have lead-in natural history, and suppose the black line is normal development, and the genetic disease, this is for like cognitive development, you have this patient that's developing less than normal, and they have a curve that we know, and we can show that we changed that curve in that individual patient. That is the idea here. Neurodegenerative disease is a little bit different. Neurodegeneration, you're losing skills. And we also know that your outcome is going to be dependent on how far along that curve of loss you're gonna be when you come into the trial. So if we get you early and you've just barely started to show symptoms, we may actually be able to normalize some things, because there may be cells, brain cells, that are kind of on the penumbra and they're not totally dead yet, but they could be resuscitated by a treatment that works. If you're further along the path, maybe we can stabilize you. If you're far along the path, maybe we can only slow disease. And so these diseases really do start at birth, and the earlier we treat them, the more likely we are to see a benefit. But the earlier we treat them, the less symptoms we have, and since drug approvals are based on symptoms, it's very hard to start out treating the patients who will benefit the most. So this is just an example of CLN2 Batten's disease and Brineura. This is FDA approved based on natural history. And so this is time to unreversed 2-point change in motor or language or both on the Hamburg scale. There was good natural history. The natural history is in red, so these people pretty much lost these skills very quickly, and Brineura prevented that in a period that was, sort of 12 or 18 months. So this was ultimately approved based on this natural history, because even the FDA couldn't stomach putting people on that red curve where they would lose skills very quickly in a placebo-controlled trial. This is another study that was mostly personally done by me, and so Adra-Betadex in Niemann-Pick type C. So the phase 2-3 trial failed. Why? Because we had non-equal randomization, and we had an 18-patient control group in a highly heterogeneous disease, and therefore the control group did not progress during the 1-year study, which made it impossible to actually have a result with the drug, because the drug-treated group also did not progress. And so we have continued I have continued expanded access at multiple sites across the USA in a 10-year study, and we then looked at the youngest patients who actually weren't eligible for the trial and asked, are we improving survival? Because it looked like we might be. And in fact, this is a study matched controls from 4 different natural history studies to this Adra-Betadex treated group of children who had disease onset very young, at below 6 years of life, and you can see there's a strong survival benefit. And this is now in as an NDA at the FDA, and hopefully they're going to agree that the natural history control group is adequate. And that drug does target the actual disease mechanism because it pulls cholesterol out of the lysosomes. Here's the Angelman studies, 3 of them. And so in the series of 10 graphs on the right, the red is natural history, the blue is the patients on the Roche ASO. Then next to it is the Ultragenyx ASO, where you can see the blue bars are going up a lot more in the same amount of time as the natural history data, which is flat for cognition. And then the Ionis data, where the pink bars are going up a lot more in the same amount of time as the black bars for cognition. So again, in this case, natural history supported the move to phase 3 and wasn't enough to get drugs approved, but currently there are 2 phase 3 studies going on and 1 about to start for all 3 of these drugs. This is a new thing that we've been doing for the last few years, an N of 1 trial in PAX1, which is a non-allele selective ASO from N. Lorem, and we're using patient as their own control, of course, for these N of 1 studies. So this is a severe neurodevelopmental disorder with seizures. The patients are mostly nonverbal. Some of them do talk. All patients have the same PAX1 mutation, which is a toxic gain-of-function mutation that messes up calcium signaling and protein trafficking in the cell, but PAX2 can compensate. So you can get rid of PAX1 and you don't have any disease, but with the mutation, you have disease. So we're knocking down, with the non-allele selective ASO, the toxic gain of function in PAX1. And you can see, this is this trajectory. We had almost 3 years of lead-in data on this patient. You can see her trajectory on the Bailey, and you can see that cognitive and receptive language starts to go up after the treatment, and that's the rate of, her rate of gain in the 6 months after treatment is in the orange bar, and the blue bars are her rate of gain before treatment. So we're hoping that continues. We have to track that long-term, obviously. But we could treat everyone with this disorder theoretically. This is our Niemann-Pick study with my expanded access with all these patients. This is just it looks complicated, but it's a simple model. We take the MPC severity score and we estimate progression from birth, which we know is an underestimate of the patient's progression because most of them haven't started progressing clinically at birth. And so we say, okay, that's their baseline rate of progression. And then we measure their progression after they get on treatment. And we subtract progression from birth from their progression on treatment. And so if the number is negative, they're progressing more slowly. And if the number is positive, they're progressing more rapidly. And you don't really need statistics. The P is significant, but you don't really need statistics to see that there are a lot more points below the line than above the line for that analysis. And that led to the company developing the drug now, Mandos, doing a more thorough analysis of that. So now this is a great example of using natural history to generate a patient as their own control design for Rett syndrome. And so this is the strategy being used by both Neurogene and Tasia, and this is the Tasia data that's shown here where natural history, that black line is the line that shows us acquisition of skills in Rett syndrome and then loss of skills and then stabilization. So basically 15 years of natural history on over 1,000 patients shows us that no one with Rett syndrome gains a developmental milestone after age 6, or it's very, very rare. So the developmental milestones that were not gained were pulled out of natural history, and there's slightly different panel of them that's being used by Neurogene and Rett, but that is the primary outcome of the gene therapy trials, where if the patient gains a developmental milestone, that is different from natural history, and therefore that can be used for registration as an outcome. So we're comparing the patient to themselves because we're only looking at things they gain that they didn't have before, and we use the natural histories data to prove that that mechanism could be used. Biomarkers are the, golden thing in any of these trials, right? The holy grail. And so this is Angelman syndrome, which shows that we have normalization of EEG delta power in both the Ultragenyx and the Roche study, as well as an improvement in sleep spindles in the Ultragenyx data from early on. And so could we use these things as surrogate measures if we actually were able to somehow correlate them with things like UBE3A levels or cognitive change? In fact, this is correlated with cognitive change, but in natural history, but we haven't shown that in trials yet. And then this is 24-hydroxycholesterol in the Niemann-Pick studies. 24-hydroxycholesterol is made in the cytoplasm of the neuron. It cannot be made if the cholesterol is stuck in the lysosome, and that's the problem in NPC. So if this runs a low level in patients who are untreated because it can't get out of the lysosome and get converted, the cholesterol can't get converted into 24-hydroxycholesterol. If you increase 24-hydroxycholesterol, you are actually changing the primary problem in the disease. This is only made in neurons, not in other cells. So we have been able to show that we are increasing 24-hydroxycholesterol as a correlate to that survival change in Niemann-Pick type C. So hopefully, ultimately, this could become a surrogate biomarker. Not yet, but hopefully soon. So my conclusions and take-home messages are we are treating genetic diseases with mechanistically and genetically targeted approaches never thought possible in the past. This will only increase, and it's going to increase exponentially, and it's very exciting that we're doing this. And sometimes We may be able to save some time with novel trial design like basket or umbrella trials. We need novel multi-domain measures and designs, which all have pros and cons, but we need to have something that is going to approach this problem. We need more emphasis on natural history as controls and patient as their own control. And I just spent the day yesterday at an Every Life meeting with the FDA trying to convince them of that. And so now is the time for implementation of all of these new approaches to trial design. We're going to hear more about that in the rest of the panels, and multi-domain measurement strategies. There's a lot of work to do, and these are our patients who are our partners in bringing these treatments to all of them as well as other people. And that's it.
All right. Thanks so much, Dr. Berry-Kravis. We've got time for maybe one question if anybody from the audience wants to come up to the mic in the center.
I guess, Dr. Berry-Kravis, if in your ideal world, how would you think of lead-in to like best design?
The lead-in?
Yeah.
Well, ideally what you would do is when you think you're going to have a gene therapy or an ASO therapy, you would immediately get the population into a lead-in study, convene the experts, come up with the best measures you can think of, and then follow those patients over time for, I mean, a lot of groups are doing 6 months, but 6 months would be the minimum. I would say at least a year. The 6 months is what the FDA has required us to do as a lead-in for N-of-1 trials within LORM now. But I think if you can follow them for a year or two while you're in drug development, that's the first thing the company needs to do, is set up that study and then work on getting all their preclinical tox and everything done. That's what I think, because that is invaluable data, and it will allow you to use a patient as their own control strategy.
Awesome. Thank you.
Thank you. Next, we have Dr. Diana Barucha-Goebel, who is the medical director at the Gene Therapy Center of Excellence at Nationwide Children's Hospital. She'll be exploring controls and outcome analysis in rare disease.
Great. All right, I'd like to thank ASGCT and the team for having this important session on just such a critical topic for rare disease trial design and overall for the rare disease community. And thank you to the session chairs for inviting me to participate in this session. I have no relevant disclosures to the topics covered in my talk today. So I'll be covering a host of kind of a few different topics, and it really segues really nicely in follow-up to Dr. Berry-Kravis's talk in thinking about some of the unique considerations really around how do we think about selecting outcomes, selecting the window to intervene for trials, and also some of the complexities that exist maybe in many diseases but become critical to statistical analysis and design, especially in the rare disease space when we're talking about smaller N clinical trials. So first, thinking about kind of outcomes as related to the therapeutic window, I'm going to talk about this in the lens of the really the neuromuscular space, but this really applies, I think, very much to many neuromuscular as well as other neurodegenerative conditions. And I'll talk about this in the lens through SMA, which I think is, really has most eloquently shown us the urgency and critical need for time to treatment. When we think about some of the clinical trials that have been conducted now, these were not the first trials for these therapies, but these were studies developed to treat SMA patients with 2 copies SMN2, those that were predicted to have the type 1 SMA, infantile onset, most severe form, for which those babies in natural history would not be expected to achieve sitting and would go on really relentlessly to progress to respiratory failure and need it for feeding assistance as well. In these clinical trials, they defined sort of pre-symptomatic or post-symptomatic infants with SMA as those less than 6 weeks of life. And treatment in that population did lead to 100% of these infants achieving sitting and respiratory independence, as well as anywhere between 71 to 80s or so percent achieving independent walking. Luckily, as we've proceeded with newborn screening implementation and now adoption by all states in the United States, we had the opportunity to identify these infants much earlier and therefore treat much earlier. And really, again, considering this a medical emergency to treat immediately upon suspect and diagnosis. And you can see that even earlier treatment under 2 weeks of life, based on the bar graphs on the top, that is based on real-world data and registry data by Cure SMA, show that there may even be a differential in percentage of patients achieving independent ambulation, even, again, within that first few weeks versus delay to treatment beyond a few weeks of life. And again, pushing the needle even further, our clinical trials now investigating prenatal delivery with therapeutics such as risdiplam. And so how do we think about this window to intervene? And this might differ for different conditions. It is not a one-size-fits-all. Are we looking at motor or function or kind of global development, development achieved, development lost, in terms of thinking about who is the right patient who may still have, who may gain from that therapy? Are we looking at histologic data? Are we looking at electrophysiologic data, neuroimaging data? And again, is there potentially room where for therapeutic gain? Perhaps the child before you has very severe motor manifestations with severe weakness and respiratory impairment, but perhaps that end organ is not degenerated with, for example, if you're talking about muscle, with severe fibrofatty replacement. So in some congenital myopathies, in other disorders of neuromuscular transmission, that window may be broader even with a very severe patient that you're caring for at the bedside. So these things need to be taken into consideration in trial design for these patients. This was already talked about a little bit, but when we think about how can we use natural history data, I think one example where this really fits very well into this is thinking about time to event, so time in terms of event-free survival or time to achieving an event. So first I'll begin showing some examples from different clinical trials that have used the event-free survival or survival overall in their gene therapy trials. So an example from ADA-SCID, a primary immune deficiency syndrome, the outcome was defined as survival or event-free survival from the requirement for hematopoietic stem cell transplantation or requirement for ERT. And in the clinical trial, they demonstrated 100% of patients with survival, 97 to 100% event-free survival at 12 months when compared to natural history data. Similarly, for spinal muscular atrophy, an outcome was defined as time to death or permanent ventilation, is defined as requiring 16 or more hours of ventilatory support, as compared to a robust natural history data from the PNCR network, really that laid the groundwork for the success in those clinical trials. Similarly, X-linked myotubular myopathy, or MTM, a systemic delivery of AAV8-based gene transfer, looked at time to ventilator hours reduction in patients who were treated compared to natural history. And in cerebral adrenal leukodystrophy, the clinical outcome was defined as survival without achieving any of 6 major functional disabilities. These included things like vision loss or cortical blindness, the requirement for tube feeding, requirement for wheelchair, complete loss of voluntary movement, and several other criteria. So again, predefined criteria driven by natural history data, and again, larger effect size in that population. So this lent itself to the use of natural history data in these clinical trials. Oops, okay. Next, when we think about time to event as milestones achieved, you can also, and again, this was also covered in the prior talk, several trials that have looked at this include studies, gene transfer for AADC deficiency, a severe pediatric movement disorder, as well as X-linked myotubular myopathy, a severe congenital myopathy. Both of these trials were able to report on percentage of patients achieving new motor milestones that, again, would not otherwise be characterized in the natural history of disease. So Again, reiterating also comments from the prior speaker. So I think we really align on kind of many of the strategies that we can think about employing, especially when you have that robust natural history data and understand the natural progression of disease. I'm next going to talk about some of the considerations around phenotypic heterogeneity and how this can impact trial design, especially in our ultra-rare diseases, where it's really critical to think about this, especially when we're trying to match patients, whether to an active control group or external natural history controls. I'm going to talk a little bit about some data from our natural history study I had conducted in my prior work at the NIH. This was in a disease called giant axonal neuropathy, a severe early childhood onset neurodegenerative disease that affects both the central and peripheral nervous system. In its classical form that you see to the left, these are young patients who have early sensory ataxia, go on to have sensory motor neuropathy, as well as will develop cerebellar dysfunction, progressive leukoencephalopathy, which you can see some signs on the brain MRIs below, and in the classic form will lose ambulation untreated by 8 to 10 years of age and go on to have additional respiratory, feeding, and other complications in the teenage years to early 20s. Conversely, there were patients that we had seen in our natural history study and that have been reported as well in other case series or case reports of patients really across the spectrum having kind of an intermediate or even a slightly later presentation or slower progression of disease, retaining independent ambulation for a longer period of time and without the leukoencephalopathy, without the cerebellar dysfunction. And so we looked at this in our cohort and subdivided the patients based on their sort of clinical impression of phenotype from this classic to intermediate to the CMT-like or later onset group. And when you do that, you take out some of that heterogeneity when you're analyzing the specific trajectory plots. And so in the top graphs here, you see one of our key endpoints that we used both in natural history as well as in the subsequent intrathecal gene transfer trial, which was the motor function measure. And also in the bottom, you see basically so these are electrophysiologic data of an upper extremity sensory nerve response. And so you can look at time to loss of that response or the retention of the response depending on the phenotypic group. And so it's important when thinking about designing the study or whether you can use that group as an external natural history control, one of the important points from the FDA is really understanding a clear, that there is a more homogeneous population for which you can compare if you're using external control, or are you thinking about how those patients are matched? And again, are you ensuring that you're not matching patients across different phenotypes, whether it's external control or in an active study with an active control arm. An example of this, where this really came into play in clinical trials, is in Duchenne muscular dystrophy. So this plot to the left here shows patients in prior natural history work of nearly 400 patients on the North Star Ambulatory Assessment, a validated scale and composites functional scale in this population. And from this data, you see these many spaghetti plots, but a best fit line that shows a mean peak age at about 6.3 years and a mean peak North Star or NSAA score of 26. But clearly, some patients are going to have a decline before that, may not achieve that peak score, or some may do better and should not show decline in those scores till much later. Subsequent work by Dr. Francesco Mentoni and others used a latent class trajectory analysis of these longitudinal trajectories to try to find patients who basically could be assigned to clusters or classes based on having a maximum posterior probability of membership or shared trajectories. And this created 4 plots or classes based on, again, these differing trajectories. Not only did they differentiate on the NSAA score, but also on other key timed functional endpoints that are used in many clinical trials in the DMD space, including rise from floor as well as 10-meter walk-run time. And how did this impact clinical trials? So in a prior study by Sarepta Therapeutics in the EMBARK study, or Study 102, this involved patients who were ages 4 to less than or equal to 7 years of age with DMD in a double-blind placebo-controlled trial with a crossover at 48 weeks. The graphs are broken up, I should have labeled better. On the left, those who were 4 to 5 years old, and on the right, those who were 6 to 7 years old. There was a statistically significant difference at the 48-week time point in those individuals who were 4 and 5 years old on the left at the endpoint at 48 weeks based on change in the NSAA score. This was not met in the 6 and 7-year-old group, but an interesting observation that you can see there with the red arrow is that there was actually a statistically significant difference at baseline between the placebo and the treated group, with the placebo being far better actually at baseline, with scores, mean scores of 24 as opposed to 19. And if you recall that plot I showed you before with where people are with their peak NSAA scores, it might suggest that, again, those same boys might have been on different trajectory plots or those clusters or classes, and that may impact what they will achieve in 48 weeks, even if you were to just follow both groups untreated. And so alternative strategies were used by the company to try to understand the efficacy in this study, especially in that population and that age group. And so there were a few considerations. One was the use of external natural history control data and propensity-weighted matching to be able to ensure that the participants you're analyzing are well matched to the natural history participants. And then another was, by leveraging natural history data, and what they did was also combining several of their treated cohorts from the clinical trials. They were able to increase and hopefully better powering those statistical analyses. And in total, that led to 50 patients in the treated group with, again, same dose across their different trials versus 83 external control patients. And this data has now been shown out to 2 years and 3 years, and you continue to show that statistically significant difference between the treated and untreated group. So there's a few things. One is, with the active placebo control group, you need to make sure that they're well controlled. Sometimes that is hard when you're doing that one-to-one allocation in the active trial, and especially in even more ultra-rare disease, that may be extremely hard to find those patients that are going to be matched to one another. But also sometimes the time to observe that clear effect, the untreated versus treated patients may diverge even more, and the effect size may be greater as time goes on. We can't have patients on control arms for 12 months, 18 months, and in some cases it's really unethical to have them all together. So we really do have to think about how we can use those external control natural history cohorts and have acceptance of them, which I'll get to in later slides. And then, some other topics that really may impact also rare disease trial design includes how do we think about the impact or influence of multisystemic manifestations? Again, I think this was also covered in the prior talk, and I'll give a few examples in the neuromuscular space. So with myotonic dystrophy type 1, this is a disease we conventionally think about muscle weakness and muscle stiffness or myotonia as the root of the disease, but also there are many other manifestations also related to skeletal and cardiac muscle health. You have risk for cardiac arrhythmias, cardiomyopathy, respiratory insufficiency, but there are also risks of involvement with vision, hearing, cognitive involvement, as well as other endocrine abnormalities. And so these also need to be, they're actively followed in our patients in clinical care. But one question is, how do we think about them in the scope of clinical trials? Fortunately, there are a few therapies now for myotonic dystrophy that are using essentially the transferrin receptor or TFR1 to help shuttling the cargo, which is either an ASO in the case of Z-basavarsin or an siRNA cargo in the case of del-desiran. But these are helping to shuttle these therapies more effectively to the skeletal and cardiac tissue, and interestingly can also cross the blood-brain barrier. And so primary endpoints in these phase 3 trials are focused on motor functional endpoints such as sit to stand, or the VHOT, which is a video hand opening time, evaluating again as related to the myotonia and the disease. And so in these, there are exploratory measures that are going to look at fatigue and cognitive function and other PROs that may address some of the other features of disease. But again, we have to think about in the future, how are we going to continue to evaluate the effects of these therapies on cardiac, cognitive, respiratory, vision, and other aspects of disease, what is treated, what may not be treated, or again, what are the benefits of these therapies across different domains or systems. Similarly, for Friedreich's ataxia, this is a disease that has onset in childhood to adolescence typically, and affects iron-sulfur cluster synthesis in the absence or dysfunction of frataxin, leading to mitochondrial iron accumulation, oxidative stress, and mitochondrial dysfunction. This affects many organs, again, classically cerebellar impairment, cardiomyopathy, but also spinal cord, peripheral nerve, in some cases endocrine abnormalities as well. So if you have a therapy, depending on, again, the route of delivery, the target, if you're anticipating more global improvement, both the cardiac disease, skeletal muscle, and cerebellar dysfunction, how are we measuring those in the clinical trial? Is one scale encompassing all? And certainly there's been a lot of work in Friedreich's ataxia with mFARS and other scales that do try to really, again, use a composite approach to track all of the components of disease in terms of motor function and coordination, but those don't necessarily have the component related to cardiomyopathy. So is there a way to think about composite endpoint or multi-component endpoints in the trial? And then you have to think about also what are you targeting when developing a therapy, and what are the goals, the needs, and the interests of the patient community as well, in targeting the heart, not targeting the heart, targeting the cerebellum, et cetera. Next, I'm going to shift a little bit and talk about how we can think about some creative trial designs when you have multiple genes for which you have an overlapping disease phenotype. And again, this may be more nuanced situations when you might be able to apply these trial design, but I think, again, it could be a nice innovative option. So an example here is from a study, it's a phase 3 study for a form of hereditary spastic paraplegia, or SPG50. This is a condition where you have involvement of the AP4 complex, and it can be caused by patients who have biallelic loss of function variants in one of any of 4 genes that encode for these subunits of the AP4 complex. There's an active clinical trial in a phase 3 trial for intrathecal gene transfer for one of those subgroups, SPG50, but leverages using the control group from some of the other genetic subgroups, such as SPG47, SPG52, and some patients who would have SPG50, allowing for a slightly larger control group, but not withholding therapy or delaying therapy in the group for which there is a product that is being evaluated. And then last few topics I'm going to try to hit on is, so I've covered this sort of throughout the talk, but just kind of honing in on the use of external natural history control cohorts. Again, I think in the rare disease space, and especially for severe neuromuscular and neurodegenerative diseases, we do have to think, about how we're designing our trials to meet what also, from a regulatory standpoint, is considered acceptable when not using sort of that gold standard of a concurrent comparator group or placebo-controlled group. And so these are points from the Guidance for Industry from the FDA. It's a 2022 document on human gene therapy for neurodegenerative diseases. And they give these criteria. I'm not going to read all of them. Obviously, the diseases we're all talking about, for the most part, have a huge unmet medical need. But other considerations in that discussion with FDA may include, again, where a placebo or concurrent control is not practical or ethical. Can the effect be, is the natural history of disease very predictable, and can the outcomes be measured or verified objectively? Especially, again, when an open label, to make sure that it's not influenced by effort or other factors. Is your treated population comparable to your external control? Can you demonstrate that? And then, again, is the effect size large enough and temporally associated with intervention to make the case that those benefits were clearly from treatment and not perhaps phenotypic variability? And so a few examples, again, we talked about the SMA, but for Zolgensma, this really was leveraged on, there was a well-defined natural history cohort and data in SMA, and a clear outcome, sitting ventilatory-free survival. So that, again, lent itself to a clear and objective finding. In metachromatic leukodystrophy, or MLD, for which there's the approval of Lenmeldy, there were concerns of having a natural history arm, a placebo-controlled arm, due to the ethics both of giving a conditional regimen that may inherently have its own toxicity in the placebo arm, and also, again, due to really a rapid progression of disease and disease severity and risks of delayed treatment. And so instead, they used a severe motor impairment-free survival as compared to, again, natural history data. And as I talked about in the case with DMD, even though there was the placebo-controlled study performed, the ability to also use the propensity weighting matching to external natural history control allowed for really a richer data set of looking at longer-term outcomes, and ultimately also it was a part in aiding in the expansion to a full approval for the ambulant patients in 2024. Finally, another consideration is N of 1 and N of few studies. I'm not going to spend too much time on this. It was already covered in our prior talk and really well covered also in the concepts of umbrella trials yesterday during the general session by Dr. Karen Musunuru from CHOP in their work that they're leading the way with urea cycle disorders. But I'm only going to highlight here just some of the concepts that are written in the FDA's so they shared a document in February of this year that was open for feedback, and I think we'll see ongoing development of this plausible mechanism framework for these individualized therapies. At the core, some concepts that are required is that you have to obviously identify the disease and really be certain of the causative impact or putative effect of that genetic variant on the mechanism of disease. Is it a targetable pathway? Do you understand or know the natural history of that disease to be able to compare to treatment in that patient? Can you demonstrate and prove or show target engagement effectively for that disease? And what are you going to look at for efficacy, whether it's clinical outcomes or biomarkers that have been established to predict clinical benefit? And there are some challenges here that, again, we can think about readiness for these, for diseases. So, do we have validated biomarkers? Is this something we can explore? And it's hard to develop these kind of for each one, but maybe as groups of diseases, we can explore those, or maybe there's, again, data already underway for groups of conditions. Another issue that may come up, though, is if there's a smaller effect size, if we're looking at slowing of disease progression and not a very large effect of survival, not survival, et cetera, you may need more robust natural history data in talking to the FDA about the use of that outcome for that disease. And then there were, again, the idea of basket trials, umbrella trials, or master protocols, where maybe you're targeting different variants of the same disease or different genes within the same kind of phenotypic group. So to conclude, overall, again, we talked about thinking about the therapeutic outcome as and the therapeutic window to explore the outcome of interest based upon your natural history data, concepts around time to event for survival or time to achieving outcomes, the impacts of phenotypic heterogeneity and multisystemic disease, and thinking about trial design concepts. As well as, really some of the guidance that's coming out from the FDA, both on external natural history control usage as well as N of 1 studies. I'd like to thank you for your attention, again, for inviting me to participate in this today. Also my team at Nationwide Children's Hospital within our neuromuscular program and Center for Gene Therapy, and my prior team and the work at NIH with Dr. Karsten Bonneman and the team on the GAN-related trials. Thank you.
If anyone has a quick question, please come to the mic.
Yeah, hi, Bill Zemsky from Connecticut. I'm a pain physician. I've been involved in efforts, some of which have been leveraged by the FDA, to get a standard outcome measure approach to this. And it seems to me, while obviously every rare disease has differences, research could be improved by developing some standard outcome recommendations, guidelines, et cetera. I don't know if any of that's going on in this world or not, but I just was curious.
So you're talking about like across diseases? Or
Yeah, across
similar to-
diseases, so that, perhaps there's a gathering of folks and expertise in rare disease who could come together, recommend some standard functional outcomes, mood outcomes, pain outcomes, et cetera, that could be then used with agreement to the FDA to move this kind of work forward.
Yeah, I think it may not be as feasible across very different diseases, but I think when you're talking about groups of conditions, if you're talking about neuropathies, if you're talking about certain muscle diseases, I think that could be leveraged again. So you're not reinventing the wheel. So you could there is acceptance on those as well. So, but.
I'm so sorry. We are over time, but please find our speakers after if they have time. And we had some clearing out, so if you're able to shift and find a seat, we'd love for you to have a seat and not be having to stand at the back. So I would like to go ahead and introduce our next speaker, Dr. Alex Fairlove. He's the Senior Director of Biostatistics at Novartis, and he'll be sharing a presentation focused on thinking beyond the clinical to decentralized trials.
Yeah, thank you. Thank you for the introduction, and it's my pleasure to be here. So I'm Alex Sverdlov. I'm a statistical lead at Novartis. So as you will see, my presentation will be focused on the statistical perspective. And so I'll talk about statistical innovation across the gene therapy development continuum. Just standard disclaimer that whatever I will present is mine, not necessarily Novartis viewpoints. And I would also like to acknowledge Dr. Avery McIntosh, who extended his invitation to me to participate in this event, and some of my research collaborators from Europe on Evidence R&D Consortium, which was a really nice collaborative effort of pharmacometricians, statisticians, neurologists, and you will see some really exciting results in this presentation. The outline. So I'll talk about what makes gene therapy special. I'll also talk about clinical development strategy and planning for success and the role of statistical scientists in this enterprise. Now I'll give an example of research innovation, the Evidence R&D program, which was a 3-year program that completed in 2025, and talk about some work in progress. So what makes cell and gene therapy special? Well, so far what I've seen, and I've been in drug development business for nearly 20 years, so it's something very special and non-standard. So first of all, gene therapies so far are one-time administration. So we need to get the dose right from the beginning. There's no redosing, there's no opportunity to change or do extensive dose finding, dose ranging studies. The source of safety signal is manifold. So safety issues can come from the delivery mechanism, from the transgene insert, from the promoter, or from over or underdosing. The standard clinical pharmacology concepts such as ADME, this is fundamentally different in gene therapy. So we're talking about biodistribution and shedding. Chemistry, manufacturing, and controls, that's another major bottleneck and challenge in gene therapies. The traditional study design, such as phase 1, 2, 3, very linear, this is not applicable in gene therapy. So the previous speakers nicely showed that there's a strong need for innovative designs that bridge together different stages of drug development. The dose finding is often constrained to just a single dose. So there's no opportunity to make a mistake. So we have to do a lot of preclinical testing and modeling and simulations to make sure that the dose that's being tested is nearly optimal. And then, of course, challenges come from the nature of the diseases, which are oftentimes orphan pediatric diseases that require novel endpoints and many other issues. So why do you need a statistician in gene therapy development? So I hope I will be able to convince you. So starting from the regulatory expectations. So this is actually the ICH E9 guidance, which is from 1999, which prescribes that the role and responsibility of the trial statistician is fundamental, and all work that is done to collect, analyze data belongs to a well-qualified and trained, experienced statistician. But that's not the main point. I think the subject matter expertise, so here you see there are multifold, multidimensional areas where statistical support can become really instrumental. So starting from the design of a clinical trial, and then doing a lot of interactions with regulators and clinical stakeholders and various other stakeholders, patient advocacy groups including, and up to the statistical analysis, modeling and simulation, and then also knowledge integration, because that's really, really important in gene therapies. And then, well, that last but not least, so there's statisticians themselves are very interested in this area. So there's a growing and increasingly deep interaction. So this is an example. That's the American Statistical Association Biopharmaceutical Section, which is called Cell and Gene Therapy Working Group. So I'm privileged to be a part of it. So you're welcome to check, but it's highly interdisciplinary. Some challenges and opportunities. So as you see, the challenge implies opportunity. And in gene therapies, there are numerous. So the previous speakers nicely indicated these challenges related to the lack of understanding of disease mechanism and lack of historical data. And sometimes we have ultra-rare diseases where there's no data at all in 1 or 2 patients. We may have some data, but very recent ones. So how do we show disease-modifying effect in that case? The very small sample sizes and limited trial duration, so they will present another major bottleneck. And then the complex interplay among the dose biodistribution response and the mechanism of action. So how do we show the target engagement and then the clinical benefit that follows? And how do we use clinical outcome measures? So that, again, just speaks to the importance of taking all these considerations holistically. I would like to highlight the important role of health authorities, the biologic centers in gene therapy regulation and approval. As of early 2023, I took this from recent publication, that there were over 3,700 cell, gene, and RNA therapeutics in clinical and preclinical development. I think now these numbers are much greater. And in the US, we have Center for Biologics, CBER, right? So it regulates all major gene therapy modalities, including vector-based products, gene-edited products, and somatic cell therapy products. So I checked, currently this site lists 47 approved cellular and gene therapy products. And in Europe, CAT regulates advanced therapy medicinal products, or ATMPs, and their website lists 32 positive draft opinions, and there were some withdrawals. And actually, you can check the website. I think there are 22 active as of December of 2025. So that's a pretty impressive statistics if we think over the decades. So the numbers are growing, and that's really good. That's the hope for the patients. So the clinical development strategy, we're planning for success. So these are 7 hallmarks of gene therapy design. This is taken from the edited volume that I listed at the bottom. So the disease-modifying or potentially curative nature of these treatment modalities. the rarity of the disease, the one-time administration, the no consensus of what constitutes the clinical endpoint and the endpoint that measures pharmacological activity, the challenges related to safety monitoring, and the ethical challenges which we should not, we should take it seriously because sometimes it's unethical to dose placebo in a trial. So that said, and just to highlight the standard clinical development plan, which is very linear, and that's something that we're accustomed to, no longer applies to gene therapy. So here, I would like to just show what a typical clinical development plan for a gene therapy would look like. So you see there's deep integration of different clinical stages. So it's no longer phase 1, 2, 3, 4, but you will see phase 1, 2 combined, seamless trial, phase 2, 3, and sometimes it's just a single pivotal trial that will be sufficient for regulatory approval. And then there are regulatory expectations that there are long-term follow-up studies, 5 years, 10 years. And then at the bottom, you will see that the natural history data plays a very fundamental role. If we want to show disease-modifying effect, we need to have a well-characterized disease progression, and then sometimes it's not really feasible because the patient registries exist for not very long period of time. So all that said, the gene therapy space presents a very, very unique and special challenge. Defining gene therapy success by pharmacology principles. So that's another nice chapter that's taken from the edited volume that I mentioned. The 3 pillars of clinical pharmacology, the exposure, binding, and downstream pharmacology define the success in this space. So next 2 slides, I have actually a mention of the existing US and European regulatory designations. And as you see, in the US, we have orphan drug designation from the preclinical stage. And then the fast track designation that gives us an opportunity to really do a faster access to FDA meetings and rolling review. Then the breakthrough designation, or RMAT, in the gene therapy equivalent. So it goes even further. It gives us some intensive FDA guidance and senior staff review. And then accelerated approval pathway, so that's something that's typically considered for the gene therapies. In Europe, EMA offers the PRIME designation, which is broadly analogous to RMAT. And then there is a conditional approval for situation where you have promising but incomplete data that lets you get on the market early, and then confirmatory study can be ongoing and running post-approval. One exciting development, I think the previous speakers mentioned, what's new in 2026, so the plausible mechanism framework. So this is something that's a genuine paradigm shift, and it just breaks the stereotype that the health authorities are very conservative, so they're at the forefront of innovation. So this is the framework that enables approval based on individual patient evidence, which is unlocking the N of 1 and N of few designs for gene therapies. And actually what it means in practice that if we have a therapy that targets a specific known genetic variant, right? So the FDA will now consider approval based on a single adequate and well-controlled study, potentially just in one patient. So that's really exciting. That's something that is completely new and has no precedence. This is still in the draft form, it should be acknowledged, but there's a link here. You see the genetic target engagement, clinical trajectory to approval. That's how the clinical development plan should look like. So let me switch gears, talk about statistical innovation, and I'll mainly talk about these 4 pillars: the efficient use of natural history data, the use of multimodal data, the improving clinical trial design, and then detecting treatment effects with multiple endpoints. So again, it all corresponds with the previous speakers' points about these challenges. So I think this just signifies that there's a lot of common thinking behind these ideas. The efficient use of natural data, natural history data. So it stands to reason that if we want to show disease-modifying treatment effect, then we really need to have a good understanding of the disease progression. So we have to have a reliable disease progression model that capture the individual patient journey over time. And then doing statistical analysis, it enables prediction of individual disease trajectories, accounting for relevant covariates and uncertainties, which is important for the design of clinical trials to demonstrate the disease-modifying effects. The use of multimodal data, so we are now in the age of data. Data is everywhere, and different data sources include clinical trial data, patient-reported outcome, data that comes from wearable devices, omics data, medical imaging, electronic health records. So this just helps us develop all sorts of things like digital twins, the joint models of different outcomes, Bayesian models, disease progression models, and all that help inform better clinical trial designs. Speaking of which, I will not spend too much time here, but just acknowledging that the master protocols such as basket, umbrella, and platform trials, they're increasingly common, and they're actually relatively novel idea. So it's about 10 years when the concept of master protocol was formally introduced, but now 10 years after that, we're using it as our toolkit, and that is really exciting. The repeated measures design, so it helps detect longitudinal signals, so just going above, just change from baseline where we would measure, quantify the difference. Now we are talking about longitudinal monitoring, decentralized monitoring, and in this space, the use of digital wearable devices, it helps, and it's really instrumental. The use of external data, that's another big topic which is actually which corresponds well with the recent FDA guidance on Bayesian modeling of clinical trials. So I mentioned it here. But it's something, it comes from the FDA, right? So multiple endpoints. So this is another important topic that's really high topic in statistical innovation. The diseases that we're considering, they're rare diseases and they're multidimensional. So there's no single outcome or no single biomarker that can quantify the treatment effect or clinical improvement. So therefore, we need to come up with some clever ways of combining data and define and validate novel endpoints that span complementary domains. And so I'll show some examples in a moment, but applying this totality of evidence statistical testing, which is going beyond a single endpoint to looking at the signals across multiple endpoints and then finding proper ways to combine this data, combine this evidence from the analysis to ensure that all these regulatory expectations such as maintenance of the type 1 error rate and so on are well controlled. So let me give an example. This is Evidence R&D, the EU-funded research and innovation program that I mentioned. So it was a 3-year collaboration, and as a result, a toolbox of complementary innovative statistical methodologies was developed to support trials in ultra-rare neurological diseases. So there were 4 work packages that I will elaborate on. So the first And yeah, the case study, this was the prototypic example, the autosomal recessive spastic ataxia Charlevoix-Saguenay, RSACS. It's a rare neurodegenerative disorder that's caused by mutation in the SAX gene, and it has progressive disability that leads to loss of ambulation and independence, and there are no approved disease-modifying therapy. We have so-called ARCA registry, which is a set of data from 990 patients, which have different genetically defined diagnoses. And the research goals were to identify the populations, develop the composite endpoints, characterize the disease progression of these diseases, and then develop statistical and pharmacometric toolkits to enable the planning of clinical trials in this indication. So the WP1, it was successful. It helped us identify subpopulations and develop optimized composite endpoints. So the established gold standard endpoint for these indications, so-called SARA score, it's a scale for assessment and rating of ataxia, and it was found to be a well-performing clinical outcome assessment. We also did the longitudinal modeling of the SARA score to quantify how fast and how differently each ataxia subtype progresses. And then there was a step forward to combine the SARA scores with digital motor measurements that come from wearable and sensor-based technologies. The second work package was to develop the disease progression model for RSACS patients, and here you see how this looks like. So this is based on the integrated data, and we used a technique called nonlinear mixed effect model to characterize the disease progression. And the model that was developed, it helps predict the changes in the total SARA score for different patients with different genetic background and different baseline characteristics. Then there were two more important tasks that were completed. So the pharmacometric framework to assess treatment efficacy and guide trial design. So this is an engine, simulation engine, that was developed to do a head-to-head comparison of various clinical trial design strategies and select one that is most fit for purpose for a given trial. And this is an example, actually, the last paper that was published. It addressed the following strategic question: which design option, which is a combination of randomization design, primary endpoint, data analysis method, would provide a valid, efficient, and robust strategy for a clinical research study to show disease-modifying treatment effect in RSACS. So this is work in progress. We continue collaboration with that group of people. Going into N of 1 and N of few trials, and that's the framework called CSC-N1, which is directly applicable and responds to the FDA's draft guidance on the plausible mechanism framework. And just one more slide to show other honorable mentions. I could not miss just to show this edited volume that I did with Dr. Avery McIntosh, who helped me get to this conference, which is at the bottom left of this, and then some other recent publications. So in conclusion, planning for success. So as we have seen from these three presentations cumulatively, the gene therapy's challenges that relate to the small populations, one-time administration, no consensus endpoints, they call for a new statistical paradigm, and they also open some doors for statistical innovation and collaboration. Interdisciplinary research is ongoing, and I'm very excited and privileged to be part of it. And I hope I was able to convince you that the statistical leadership in gene therapy program is not optional. So it's something that's really essential to ensure valid evidence, regulatory success, and ultimately patient access to potentially curative therapies. And thank you very much.
Thanks so much. We can take probably one question if anyone's got it from and wants to come up to the center mic.
Alex, can you talk about nonlinear modeling for diseases where we don't exactly know how change over time is going to have? I see you did like a sigmoid curve, but from a statistical perspective, what do you say is like the correct model?
There is no, I'm afraid I will not be able to give a unifying answer. As you see here, this graph shows the complexity. So we have bits and pieces of individual data, and we want to develop a population model that helps you predict individual trajectories depending on the baseline covariates and some other predictors. So it requires data. Without data, there is little hope, and that's why you see I avoided to mention any other techniques like machine learning techniques, which are heavily data driven. So this is more statistical, nonlinear mixed effects model, but it still requires data. So in this case, data came from 990 patients, which were a big blessing. But as you see here, for instance, there are two plots that show the sparsity of the data for some particular subgroups. That's always a challenge.
Thank you.
That's a great question. Thank you.
Next, we have Ms. Julie Tierney, principal at Levitt Partners and former senior FDA leader. She will be going through best practices for engaging regulators for your rare disease trial.
Hi, I'm not as tall as he is. Great. Thanks so much for having me, and really interesting talk from the previous speakers. My slides are going to be shorter, so if you had questions that you were holding for the other speakers, we'll have time, I think. But I'm going to talk about best practices for engaging regulators for your rare disease trial. Prior to Levitt, I was at FDA for almost 17 years in a number of different positions, including deputy director at the Center for Biologics and chief of staff. I am an attorney by training. Great. What I'd like to do, though, is talk about sort of the policy and regulatory environment at FDA, including some recent developments, starting with sort of a level set of what we're dealing with, which is sort of unprecedented times. Then talk about opportunities for formal engagements around rare disease development programs, including expedited programs and opportunities to discuss things like novel endpoints with FDA to gain acceptance of those. Turn to some more recent developments, including the plausible mechanism framework that some of the other speakers have mentioned. And then finally, opportunities for engagement with the agency outside of specific development programs. So just starting, we've got, just to level set, it's been an unprecedented year at FDA, I think, with a lot of changes, unprecedented week with a lot of changes. We have a new acting commissioner at the agency who comes from the foods program. And at the same time, we have acting leadership throughout the Center for Drugs and Center for Biologics, both the directors, and there's a number of acting directors within CBER. So that's led to some uncertainty. And of course, everyone's aware over the past year there's been a large exodus of staff from the agency. A fifth of the agency is left, so that means that you've lost a lot of experienced reviewers as well as leaders, and that the remaining staff are under tremendous pressure to meet deadlines and fill in for their colleagues who are no longer there. At the same time, we've got unexpected changes in FDA positions on development programs over the past year, with a number of complete response letters that have been issued that sponsors have sort of been taken by surprise on FDA's position. Perhaps with Dr. Prasad leaving, that might turn around. And then we also have policy announcements that are coming out in very unexpected ways via, journal articles, speeches, podcasts. And so it's sort of hard to keep track of it all. I think, though, thinking about engagement with FDA through all of this, I think, one, really important to take opportunities to educate FDA during development, educate about the disease, about trial designs. As I mentioned, folks are under a lot of pressure and may have lost some of their experienced colleagues. And to the extent that you can present clearly, communicate clearly with review teams and educate them, I think it's really important. Also very important to carefully document agreements that are reached on path forward, including a special protocol assessment agreements if needed, just that you may have to turn back to it, and you want to make sure that there's good documentation. I think really important to be respectful and collaborative with the review teams. These folks are likely to be at the agency after the political leadership is gone. If you have a pipeline in a particular area, you're going to be dealing with them on a repeat basis. So I think important to be respectful and know that they are under tremendous pressures. At the same time, I think if needed, challenge review teams, advocate for a fit-for-purpose strategy that's supported by data, bring the data and the scientific justification. And I think there's different strategies for elevating if you don't feel like you've reached resolution, and that's going to vary by sponsors' risk tolerance, by the review team interactions, and sort of the overall FDA environment, which of course is changing by the day. So just a rundown of some of the formal opportunities for sponsors to meet with FDA. Of course, I think early interactions are always a very good idea, and the interact meeting types that were developed within CBER have extended to both CDER and CDER. So that's like a pre-IND meeting, and really can help to make sure your development program is as efficient as possible. But you need to make sure that you are ready for that and have your proof of concept in line. Type B meetings are milestone driven and really opportunities to talk with FDA as you move between the stages. Type D meetings are going to be follow-up meetings on particular issues. Type A meetings are coming up with the complete response letters where you've got programs that are stalled. And so those are specific type. And then, of course, Type C is anything that doesn't fall in the others. But in particular, under the most recent round of PDUFA commitments, there's Type C meetings for novel surrogate endpoints that can be used as well. As we think about early engagement being so important, FDA's expedited programs, which the other speaker referenced as well, are really an opportunity for FDA to engage earlier and more frequently with sponsors that have programs for serious diseases that are addressing unmet needs. So fast track designation, breakthrough therapy designation, regenerative medicine advanced therapy designation for cell and gene therapies. These are all opportunities if the criteria is met and sort of increased amount of interactions. I think one of the benefits of breakthrough and regenerative medicine that are above the fast track is sort of the senior leader engagement, as well as meetings specifically around endpoints that are called out in the RMAT statutory provisions. And then finally, once you get to the review stage, hopefully those early interactions have helped to make sure that everyone is educated about the development program, that the sponsor understands what FDA is looking for. I think as we have these scientific technologies, these advances in science and medicine, and the ability to develop products so quickly, and if you've got these if you have the expedited programs where you're getting these increased interactions in your clinical development program, hopefully is accelerating. It's really important to think about CMC readiness, and of course that can be a stumbling point for cell and gene therapies. a few weeks ago, FDA put out a chemistry manufacturing controls flexibility guidance that summarizes the flexibilities that they're willing to entertain and kind of puts in guidance form something that had been on FDA's website in January. But my understanding is that there are still issues that are really preventing development from moving forward. So priority review is going to bring in a product for a 6-month review rather than 10 months. And in these rare diseases, of course, every minute counts. And accelerated approval, allowing for approval based on a surrogate endpoint that's reasonably likely to predict clinical benefit or an intermediate clinical endpoint. And then again, hopefully, if you're able to identify that surrogate endpoint, it's going to bring a product to market sooner than if you waited for clinical endpoints to develop. Right. And so in rare disease trials, of course, the other speakers talked about sort of getting FDA to accept novel endpoints, novel surrogate endpoints, and that certainly is a challenge. I think the early engagement with FDA through these Type C meetings for early consultation on novel surrogate endpoints, including scientific justification. I think many disease groups can tell you, though, it's sort of a long process with FDA to get them to accept novel endpoints. Again, as I mentioned, there are meetings specific to endpoints that are provided under the RMAT designation. The Rare Disease Endpoint Advancement Pilot, the RDEA pilot under this round of PDUFA, is another opportunity. I think it perhaps has not been utilized as much. It puts you sort of in a special program where there's more attention and development of the novel endpoint. But there are disclosure provisions, which some folks are not comfortable with, which I understand have sort of decreased its uptake. And then finally, there's a drug development tools qualification program that was put in place as part of 21st Century Cures that can be used to qualify an outcome measure or biomarker across different programs so that it can be utilized across programs. Unfortunately, that hasn't seen the uptake that we hoped for when we put it into law. Worked on that provision with fewer drug development tools qualified than I think we'd hoped for. Friends of Cancer Research did a paper on this recently, and I think the other concern that folks have with it is if biomarker is qualified, often the review division is not accepting that biomarker. So it's sort of not operating as optimally as hoped. So I want to turn to some more recent developments that are also, opportunities perhaps. So we've got this Commissioner's National Priority Review Voucher Pilot Program. We will see whether it stays in place after the commissioner has left. It was framed as a pilot, and I think it's up to like 20 different products. It was supposed to be 5. And it's really supposed to take products that align with national priorities. And in the rare disease space, I think, thinking about, potentially curative products, the recent Regeneron gene therapy for deafness, but also thinking about affordability and onshoring. Those are also issues that have led to vouchers. So this is targeting review in 1 to 2 months via a tumor-style review board. There have been a lot of concerns raised about the legality of the program, whether or not it will be durable, as well as it sort of allowing for political interference in the review process. You can imagine if you're a review team and you go into the commissioner's suite and face your boss's boss who have opinions on whether or not a product should be approved, that could potentially interfere with your independence. Great. And then I would just also say that receiving a voucher is not a guarantee of approval. I think we had thought when it was first announced that these would be they picked the winners, pre-baked them, but it does seem that even some of the products that have been selected have not been approved in the end, such as the disc medicine product for porphyria. Another program I just wanted to highlight is the Cedar Rare Disease Evidence Principles that was launched out of the Rare Disease Innovation Hub, which is a cross-center rare disease program. It's really focusing on very small patient populations where the therapy is specific to the correction of a genetic defect. And it's trying to, I think, give sponsors an opportunity to come in early and get sort of a shared agreement on the path forward. And I think one of the benefits of the program is engagement of the CBER-CDER Rare Disease Policy and Portfolio Committee that's part of the hub. So you've got senior leaders from across the centers and hopefully alignment across the centers on the path forward. And it really manifests a commitment by FDA that the review will encompass additional supportive data, in particular, one adequate and well-controlled trial plus confirmatory evidence as the way of meeting the substantial evidence bar. And of course, the commissioner and Dr. Prasad had issued a New England Journal article a few months ago saying that one adequate and well-controlled trial would be the default standard moving forward. That's not really news in the rare disease space, although it's been somewhat unevenly implemented. I think most rare disease and oncology approvals have been based on one adequate and well-controlled trial plus confirmatory evidence versus the two adequate and well-controlled trials that you see for something like diabetes or cardiovascular disease. Again, these sort of unusual ways of making policy that was issued in a journal article behind a paywall, and I understand that guidance has been updated and is with the White House for review to sort of make that a more durable policy. So we'll be looking for that. The other speakers also mentioned the plausible mechanism framework. I think that there's still a good number of questions to be answered about how that'll be operationalized as a regulatory matter, including its scope, how sponsors can engage, because I think here again, early engagement is going to be really important. And you heard the team from CHOP talk about the Interact meetings and pre-IND meetings they had that made the process for developing the product for baby KJ so smooth, because they knew what FDA was going to be asking for when they submitted the single patient IND. FDA knew what they should be expecting, and they were able to move through that quickly. So I think that's important as FDA works to finalize it, and perhaps there's some details that they will need to work out. On that, sort of related to that, though, this idea of leveraging knowledge, I think it's really important as we think about individualized therapies, N of one, N of few, to think about what we're leveraging across those products in terms of the backbone, and what we can not recreate the wheel each time, so that there can be some efficiencies there. So we've got the platform technology designation that was enacted in 2022. I think it's based on an approved product, so you can't get the platform technology designation until you have a product approved. So that's a real limitation, I think, for some developers. It's the same sponsor can rely upon data from about the platform, which is something you've always been able to do, but I think it just sort of codifies that ability. But I think importantly, that's platform with a big P, but the sort of the spirit of the platform designation, the spirit of leveraging platforms is really going to be in this guidance, leveraging prior knowledge for cell and gene therapy that CBER's putting out we expected by the end of September. And I think that'll be really important to help move forward the plausible mechanism framework, but just also thinking about if you have the same platform, you have the same backbone, vector, what can you leverage preclinical, CMC, safety information that can help move forward programs for very small populations more quickly. Great. And then, just thinking about outside of an individual product development, how to educate FDA about a disease, about patient perspectives. I think the patient-focused drug development program has been in place for many years, has really worked to move from sort of anecdotes to more scientifically based approaches. At the same time, I think the intent was to humanize the review process for reviewers so that really understand what's important to patients. So there's the patient-focused drug development meetings, both FDA initiated as well as there's a variety of externally led patient-focused drug development meetings. And I can say that FDA reviewers really do pay attention to that and try to include that as they're thinking about their review decisions. Their patient listening sessions that are led out of the patient affairs staff in the commissioner's office, again, an opportunity for patients to come in and talk about what's important to them. Critical path innovation meetings, the Center for Drugs has that program, and often they will include teams from the Center for Biologics, and those are really about non-application specific drug development issues. So oftentimes patient advocacy groups have come in, or consortia have come in to update FDA. And so it's an opportunity outside of the development process to help educate reviewers. The Rare Disease Innovation Hub is also a really important resource, not just for patient groups, but for researchers and industry. The hub is not going to get involved in specific review decisions, but I think one of its primary goals is to make sure that the centers are aligned in terms of the advice that they're giving to similarly situated programs, as well as make sure that the external feedback is making its way into the FDA ecosystem and acted on in a meaningful way. So not just listening, but exchanging information. And finally, this science-focused drug development meetings has been a proposal that's been kicking around for the past 1 or 2 years, building on the patient-focused drug development meetings, but this time sort of bringing together patients, researchers, industry, and FDA to really dig down deep on some of these issues around particular diseases and outcome measures. I've been to a few that were not officially titled that, but I found the dialogue and engagement there to be very helpful. Ultimately, we want to make sure that FDA is aware and educated about what's happening outside because they are under a lot of strain, and at the same time, make sure that the work that's happening is fit for purpose and will ultimately support approvals. So, great. All right, that's it.
Please come up to the mic. In the meantime, while waiting questions, if you're interested in connecting with patient advocate leaders and those interested in supporting rare disease drug development, please join us during the patient advocate meetup taking place later today at ASGCT Central in the exhibit hall from 3 to 3:30, booth number 1463. Oh.
I guess we'll just let it in.
Okay. All right. Thank you.
I don't think she wanted to.
She didn't want to.
I don't think so. That's all right. My boss came.
What's that?
My boss came.
Oh, wow. Okay.
Yeah, he was right there in the bald head walking away. I can't believe how well attended it is.
I know. Like, yeah, now everyone has. That's
Yeah,
next
they're
slide.
just gonna grab everybody.
Okay. It's been busy this conference.
Oh my god, me too. Oh. Hi. 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 Good morning.
For my non-profit organization called the Dream. So we're currently doing studies for studies and studies for single patients. So I'd like to be involved.
Oh. That's a good question. Let me come down there because I think these mics are still on. Sorry. Okay, I refilled my water.